Hello, I'm David Tabb. I am a bioinformaticist working over at the University Medical Center of Groningen. What I'd like to do today, though, is to present some slides that we assembled for the OpenMS South Africa Roadshow earlier this year. The South Africa Roadshow this year featured a lot of plant biotechnology and also an emphasis on metabolomics that's been missing from some of my earlier slides. So I would start by saying we're going to focus on two very different kinds of strategies analytically for interpreting metabolites. So can we target particular classes, particular molecules that we think are potential biomarkers, or can we perform discovery experiments that are untargeted, that are allowing us to take a survey of all the different metabolites available in a particular sample type? So we need to start, I think, with a bit of an overview of metabolomics. Metabolomics can mean many things to many people, and I think talking about some of the ground rules for what the terms are going to mean can be useful. And from there, of course, we want to spend a lot of time differentiating targeted and untargeted, but we see a lot of different terms used for these technologies. So discovery methods or verification methods are also common in biomarker studies from proteomics. So I'm going to try to explain these with references between the two. And from there we need to talk a little bit about the different separation technologies that exist for it and what analysis technologies are used to produce the data for identification or quantitation. And at the very end of the talk I'd like to say just a little bit about repositories and standards. So we can start with the map of all processes, all biological processes taking place within an organism. That's obviously of huge complexity. Metabolomics typically subsets that down a little bit and begins focusing on chemical processes of a particular class. So you might find that somebody is studying carbohydrates particularly, or someone is studying lipids particularly. In general, metabolomics has to choose what it's going to emphasize for a particular study. But the metabolism as a whole is a much larger space of things. We have different biological processes that involve anabolic processes where we want to build up something like when plants photosynthesize to produce glucose. That's an anabolic process of putting these carbons together to make the 6 carbon sugars. And catabolic processes where we destroy something to produce energy. So the process of fermentation or the process of respiration produces energy stored in the form of ATP by breaking down larger molecules. Okay, so we often then think when we're examining one particular metabolic process that perhaps we have an enzyme involved that is accepting edux, these are the inputs to this chemical reaction, and products that are the outputs from that reaction. So taken at an individual reaction, the edux and products are going to have this balance that's driven by what enzymes are present, how they're activated, etc. Okay, so we're not really going to be spending our time talking about enzymes, unusually, because this is not a lecture about proteomics. But it's important, I think, to remember that individual biochemical reactions do not exist all by themselves in the system. Generally, there's a whole cascade of different enzymes that are shepherding the edux into products, and then those products serve as edux for the next reaction. And along the way, we get a metabolic pathway. Or you may find many branching pathways that come together at various points or separate at different points and treat that as a biological network. Now, the metabolome and the proteome have very different problems to solve. So we'll start by saying that although it's pretty straightforward today to produce an annotated genome for a species, we can then say this set of protein sequences is possible for the central dogma to take place upon that we have the transcription of this gene. Those transcripts then get shuffled, and then those shuffles, those mature messenger RNAs get translated to produce protein products That's all reasonably straightforward. So you can say effectively what is the complete catalog of molecules that can be produced by the ribosomes of an organism. Now, things get a little different when we talk about metabolites, because it's not always that apparent exactly which metabolites are possible for a given organism. And remember that these slides were produced in the first place about plant biotechnology, and plants, as nature's factories, have amazingly complex workflows that can produce all kinds of interesting outputs. So we can certainly have tens of thousands of different types of metabolites that can be formed. And frankly, if you start thinking about, say, a triacylglyceride, you've got 3 different fatty acids attached to a backbone. If you stop to consider every permutation of possible fatty acids across those 3 sites, and every possible double bond, a point of unsaturation in those fatty acids, certainly you can get into thousands just talking about lipids. So metabolite databases, if formed virtually, can contain easily tens of thousands of molecules, and if you have something like a spectral library accompanying it, at least some of those structures will be paired with a prediction of what ions should be seen if you were to fragment this metabolite. Okay, so let's talk then about this difference between metabolome and proteome here. We start then that metabolites come from a very wide diversity of chemical classes. To talk about what a lipid structure looks like, or what a sugar looks like, or what a free amino acid looks like, are all very different chemistries. Yes, you do have things like condensation reactions are used to attach different compounds together, and that applies in a very wide diversity of structures. But just generally, the types of chemistries embodied within a metabolite can vary tremendously. And when we start thinking about that from the world of proteomes, well, the proteomes are all chains of amino acids at base. Even if you start getting into something relatively unusual, like say a cyclic peptide, you're still talking about peptide bonds, you're still talking about side chains, and so on. But the type of chemistry encoded in a metabolite structure is quite a bit more complex than that. Similarly, when you're trying to explain what protein you've seen or what peptide you've seen, you can generally give the shortcut of simply saying, well, it's a L followed by a V followed by an A, representing the single amino acid codes for that sequence in the case of a proteome. And yes, if you're going to start adding in things like phosphotyrosine or whatever, then you need to involve PTM definitions. But generally speaking, that's a pretty straightforward way to communicate what you've got. When you're trying to describe the structures you've seen in a metabolome, on the other hand, you're probably going to need to describe the structural formulae that give rise to that structure. And then finally we get down to the masses of the things themselves. Metabolites are generally quite light. A metabolite of 300 Daltons is not uncommon, and yet when we start thinking about proteins, we're talking about something much, much heavier than that. Just an individual amino acid is usually an average of something like, that's the cat dashing by, 110 Daltons for an individual amino acid. So you can see that even if you connect together 3 amino acids, you've built a very small peptide, but you're already larger than most metabolites. So when we start trying to measure these metabolites, we may have a very broad question that we're trying to address, or we might have a very focused one that we wish to address. So with targeted metabolomics, we usually have a very clear notion of which molecule or molecules we want to study. So maybe we're studying ascorbate in blood, something like that. So we probably have some very clear notion of how the intact molecule behaves. So we would know things like what its retention time is likely to be in liquid chromatography. We would know what its intact mass should be, what ionic charge states it'll adopt, and probably, if it fragments, what fragment ion mass to charge values should be produced from it. With that kind of specific detail, it becomes possible to quantify a metabolite very accurately. And that is what we use in targeted metabolomics. Specifically, you may want to target as many as 100 different metabolites in a single LC-MS experiment. So for that, targeted metabolomics is going to require that we have a predefined list of precursor ions that we want to look for, and often a predefined list of which fragments to expect for them as well. That's quite different when we get down to untargeted or broad metabolomic profiling. In a case like this, we already have to make some choices. You're not going to be studying lipids at the same time as you're studying small polar molecules. These are they have quite different behaviors in separation and quite different behavior in mass spectrometry as well. But it may be that the answer you're looking for lies within the world of lipids, for example, or within the world of sterols, something like that. And for that purpose, we would be able to know what type of separation chemistry will be compatible with all these different metabolites and so on. But we still have the problem of being able to identify that. We'll spend some time talking about that in a different lecture. Okay, so the untargeted or non-targeted discovery experiment is to say what metabolites are here. The targeted is much more about quantifying some in specific. Now, before we move much further, I want to stress that lipidomics in particular has really matured as its own branch of metabolomics, and there are a lot of things about it that sort of differentiate it. So I'd like to talk about what's feasible here. I've looked over just a couple relatively recent publications, and yes, we do see things like nanospray, LC-MS being used for identifying hundreds of lipids in a single experiment. And you can do that without prior fractionation. So basically, each sample leads to one LC-MS experiment, and now we can identify hundreds of lipids in just that one shot. So this is not perhaps as complex a mixture as what you'd see in a whole cell lysate that's been digested to peptides. But in this case, you can still identify quite a few compounds just using roughly the same equipment as what you would see in a proteomics experiment. It is also possible to use strategies like database search for identifying these phospholipids. There are really good models that explain what fragment ions you should expect to see in positive ion mode or negative ion mode, typically negative ion mode in lipid mass spec, to say which fragments we expect if it were this particular peptide. So some of the techniques we know from database searching of peptides have parallels as we move over into the metabolomic space. On the other hand, something that really differentiates lipid mass spectrometry from that of, say, carbohydrates or something like that, is that the molecular size we're talking about tends to be much, much larger. By the time you get to something like cardiolipins, you can have lipids large enough that they have a huge diversity of possible decorations with fatty acids, and you can get a mass that's even above 1,000 Daltons, which is, I would say, pretty distinct from what we expect in most metabolomic studies. So I would point you to a resource put together in mostly human samples. The Lipid Maps Consortium has put together some really amazing work. And you can see that their database catalogs more than 50,000 distinct lipids. So we're in a different kettle of fish when it comes to how many distinct molecules are possible. These things are not wholly different from each other. We're saying that if you have the same glycerol that's been decorated by 16 carbon fatty acids rather than 18 carbon fatty acids, that's a different species, right? So that's going to cause the numbers to climb quite rapidly here. But their structure database will index that for us. But even if we look just within the realm of lipids, we see that fatty acyls, glycerolipids, glycerophospholipids, sphingolipids, sterol lipids, prenol-lipids, saccharolipids, and polyketides are all different divisions within this structure database. So there's a great diversity of molecular types that appear just in the world of lipids. So we often speak of lipidomics as, of course, included in metabolomics, but it has its own methods and its own community, so it's worth thinking about it a bit separately as well. All right, now I said we were going to talk a little bit about the analytical chemistry of this as well. And so I would like to bring us to the world of proteomics for just a moment to bring in some parallels. So untargeted data-independent acquisition versus targeted selected reaction monitoring. These are two different types of mass spectrometry methods that we typically use to quantify stuff in proteomics. But these are a good parallel, I think, to explain this difference between untargeted and targeted experiments. So let's start with the data-independent acquisition experiment. Here, we're probably going to try to quantify as many peptides as possible from a mixture. And you can do this with more than 10,000 peptides being quantified in each experiment. So in this case, we're going to combine MS1 scans and MS2 scans, so tandem mass spectra with full scan mass spectra, much as we would in a classical DDA style experiment, a data-dependent acquisition experiment. But we're going to tell the instrument to regularly pull these isolation windows of particular peptide ions. So here I've got a design borrowed from Lindsay Pino's paper here in 2020 that we've got 24 mass unit wide isolation windows, and we're going to co-fragment everything in that range with a precursor mass in that range, and then look at all the fragments that result from that co-fragmentation. So here we have each of these windows. There are 25 of these windows spanning the space between 400 mass to charge and 1,000 mass to charge. So this is the range of peptide ions that are going to be measured here. And each of these fragment ions will get measured roughly every 3 seconds, which allows us to build a chromatogram for not just the peptide ions in intact form, but also the fragment ions that are seen from them. So this gives us the ability to quantify a huge number of peptides in a single LC-MS-MS experiment. Now that's quite different. This is not tuned to a set of 100 peptides we think are important. We're going to quantify tens of thousands of peptides in these experiments. That's very different than a selected reaction monitoring experiment, an SRM. In this case, we're going to generate things called transitions, which is to say a peptide that's on our target list has this mass, and when we blow it apart, it's going to create fragments of these masses. So each combination of an intact peptide and fragment ion mass charge value is called a transition. We're going to have a huge pile of these transitions, and we're going to keep cycling through them, monitoring them all over the course of this experiment. That gives us the ability to quantify this tight set of target peptides very, very accurately. So these are both quantitative experiments, but the DIA experiment is trying to do usable quantities for a huge number of peptides, and selected reaction monitoring is trying to produce very accurate quantities for just the peptide ions on the list. So in proteomics, we would think of this as a discovery experiment or untargeted, and this as a verification experiment or a validation experiment. This is something where SRM is targeting just a set of pre-specified peptides. So it seems lovely that if you can quantify 10,000 peptides, isn't that everything you would ever need? But that's not really true. Looking at a great diversity of ions comes at a big cost. Calibration is and we'll talk about what this cost is as we go through the slide. We also need to be able to turn intensity values that have been measured on the mass spec into quantities if we're really going for a tight quantitative experiment. So let's talk about this diversity coming at a cost. If we look at a pure discovery, untargeted experiment, we are able to measure a huge number of metabolites, and yet the reliability of the quantification for it is likely to be pretty poor. If we are able to compare signals across a whole class of metabolites, that's pretty good for us to improve our quantitation, to be able to say that we have at least some expectation of which of these lipids is playing a larger role in this sample. But ideally, we would have some isotope labeled internal samples. If we have these labeled standards internal to the mixture, and we know how much of the standard is there, when we measure the experimental version of that peptide, we can compare their intensities to say, oh well, there's twice as much of this ion present in the mixture experimentally than appears to be true from than we spiked into it in this labeled standard. So as we get further and further down the scale, we're getting better and better at quantifying, but the number of ions that we can consider at once is shrinking. So maybe we're starting with 1,000 different metabolites that we're trying to quantify as well as we can en masse, but by the end we probably have one or just a handful of ions that we're trying to quantify very, very accurately. And for these, we're going to need internal standards and a calibration curve. So being able to know experimentally that when I spike this much into the sample, I get this much intensity in response, and as I increase that by several fold, I can see where the range of linearity is. I can say, I know that I have very good quantitative performance on this instrument if I have between this concentration and that concentration. But if I have too much of it, well, I get an attenuation of the signal up against the maximum intensity that can reasonably be measured by the instrument. And if I have too little of this sample spiked in, I can't even detect it, or I certainly can't quantify it at that level. So as we bridge this transition, this spectrum basically, moving from untargeted to targeted, we're raising the standards we require for a successful quantitation substantially, and we get much better at saying, well, it's up 20%. I mean, if you have internal standards that allow you to say that this metabolite's apparent increase of 20% is real, that means you're doing this with internal standards and so on. You're not going to be able to get that kind of information from a very broad look at all possible metabolites in the mixture. And that brings us to kind of the difference in how these kinds of experiments look from a high level. So let's start over here with an untargeted experiment. Typically at this stage, we are trying to evaluate what metabolites exist in the mixture altogether. So this comprehensive profiling means that we're going to be starting with selecting an untargeted method that will tell us about compounds of this type. Not just like metabolites, but I mean a particular subclass of metabolites that are of particular interest in this case. The extraction processes we use are not specific to one metabolite or a handful. The extraction processes need to give us a good cross-section of all the molecules of that type. We can do our LC-MS. We generate extracted ion chromatograms. We need to align those chromatograms to be able to do relative quantities and so on. We also need to be able to understand how much the chromatography has shifted from one experiment to the next. That gives us retention time correction. And finally, we have some fairly complex multivariate statistics that allow us to put together a list of metabolites we think are particularly associated with our phenotype. In the clinical biomarker space, you would say these are our putative biomarkers. Now, the information from this untargeted experiment is then transmitted to this next round. We are informed by our broad look at metabolites to say, this subset of lipids is of particular interest to us. We want to quantify these well. So this gives us a targeted metabolomics workflow and an extensive methods, analytical methods optimization. We're no longer trying to tell information about every molecule in this class. Now we have the specific need of quantifying just this subset of putative biomarkers. Okay, so we're going to need standard metabolites, which is to say chemicals we can spike in that allow us to quantify these. We're going to need to improve the way we do LC-MS to give us the very best possible separation of these metabolites. We're going to do a selected reaction monitoring style experiment, one that brings together knowledge of which metabolite ions are we looking for, what are their masses in other words, and what fragments result from them. we're going to need to develop standard curves to be able to quantify these. And eventually, we're going to have a much simpler story when it comes time to statistically analyze these data, because now we have much more accurate measurements of their quantities, and it's a relatively small handful of putative biomarkers, not just everything in that whole molecular class. So, is it likely then that everything in metabolomics should be analyzed by LC-MS-MS? I mean, yes, that's where I live, but that's not to say this is true for the whole field of metabolomics. In practice, the field of metabolomics has done a lot of work with gas chromatography, and I'll have a separate lecture about that filmed shortly. Yes, you can do a whole lot of valuable things with liquid chromatography, but there are certainly kinds of compounds that you can really not deal with by liquid chromatography. And if you can't do it by GC, you're probably going to need to use something like capillary electrophoresis. But these three methods have different trade-offs, and different subsets of these compounds can be found by those three methods. So let's talk through them. Gas chromatography mass spec, GCMS, is extremely widely used for metabolomics. Now, GCMS typically uses something sometimes called electron impact dissociation, and EI is really good if you're trying to do identification of metabolites, because there are great big libraries, spectral libraries, showing what a particular metabolite looks like when it's been through GCMS. But generally speaking, you're going to have to use some sort of derivatization to cause many molecules to be volatile enough that they can be ionized by electron impact and separated in GC as well. So generally speaking, we've got relatively low mass ions that often require some sort of derivatization to make them volatile, to put them into gas phase. Okay, next up, liquid chromatography mass spec. Yes, this is very, very widely used in proteomics, but it has a very good usage pattern in metabolomics as well. So the derivatization that's necessary for a lot of GC work is not really necessary here. Typically, the samples in a physiological environment can be introduced to the liquid chromatography environment without a lot of extra work. But there are some problems when it comes time to quantify, because all the different molecules that are coming out of chromatography are competing for which of them is going to ionize most successfully. And you might be co-eluting with another metabolite that's really good at grabbing protons or very good at giving up charge. So in that case, our quantitative accuracy can be compromised in LC-MS if a whole lot of ions are coming off the column at the same time. They tend to crosstalk a little bit in that respect. Now, capillary electrophoresis is still, I think of as sort of a relatively, it's a relatively technical separation type that requires specialized skills to make it work very, very readily. So it's not, I would say, as widespread as LC-MS, and certainly not as widespread as GCMS. But it's really great if you're working with polar and charged species. On the flip side, it's very much something that can only deal with very small sample volumes. So you're going to need to make sure that your mass spectrometry is very well tuned to be able to deal with very small small numbers of molecules for each metabolite. And I think it's worth saying that not everybody working in metabolomics feels like mass spectrometry is the right way to go at all. There are plenty of people doing 2D NMR. So this is a brilliant technique. It's very fast. It's non-destructive, meaning you get your sample back. But the number of components that you can tease out of a sample is typically going to be 200 or less. So if you've got the desire to see 1,000 different metabolites from your experiment, I think that NMR is not likely to give you that. Now remember, this talk was originally written for plant biotechnology, and I think it's worthwhile talking about what makes plant metabolomics distinct from more general applications of it. So I found a couple papers on this, and this is kind of two different ways to look at this problem. So we start with responses to stress. If you stress out a grad student, you might find that they run away, right? Plants don't have that opportunity. So when you stress a plant, they need to be able to deal with the problem in loco. So we can find that signaling compounds used in plants can be of unusual diversity. Secondary messengers can be very interesting to understand how signals are transmitted through plant cells. Phytohormones are interesting because the plant needs to be able to send different signals from one part of the plant to another. Defensive and protective chemicals. Certainly there are lots of plants that make use of chemistry to send away obnoxious animals that might eat the plant, for example. So there's plenty of information there. The second paper I looked at spelled out 4 different pathways that they thought were really essential to understanding plant metabolites specifically. They said that polyketides and fatty acid derivatives were really special to the metabolomics of plants, as were terpenoids, alkaloids, and phenolics. Certainly, anyone who's enjoying a fresh cup of tea is probably thinking about which lovely phenols are producing that wonderful buzz on the tongue. So we owe plants an awful lot for the diversity of the molecules that they produce, and they are a very interesting place to pursue metabolites. Now, I said I was going to talk just a little bit about repositories and standards. I have just the one slide to discuss that. As part of the proteomics community, I've been very, very grateful for a service called Proteome Exchange, which allows all the different repositories around the earth who work in the space of proteomics to have a shared index system and even to mirror files from some of the other repositories that enable them to download a data set corresponding to a paper. I read a paper in proteomics, there's a PXD number, I look up the data set, I download it. It's great. Something very similar existed in in the metabolomic world. That's called Metabolome Exchange. But the funding for Metabolome Exchange was never as secure as it was for Proteome Exchange. As a result, today, if you go to the Metabolome Exchange website, you'll see a nice thank you message expressing the gratitude that you came to them. However, they don't have the payroll to have people staffing this site regularly. So Metabolome Exchange appears to be in a defunct state, and I really, really hope that they get the resources they need to continue this service. There are a number of repositories out there for metabolomics. The Metabolomics Workbench, MetaboLights with a GH in it. GNPS is a really valuable one in natural products over at UCSD. And MassBank Post. All of these are still up and running, doing good work. There are, however, a couple of maybe less well-known repositories that are serving the plant community. So Plant Metabolome Repository and Mary B are both out there, and you should be aware of those if you're trying to look for a metabolome specific to your plant of interest. The Metabolomics Standards Initiative is something I really applaud. I'm part of the Human Proteomics Organization Proteomic Standards Initiative, and the MSI is our counterpart for the metabolomic world. It seems like they've had a brilliant start, but they're kind of in the same way as Metabolome Exchange, trying to find their way again. The SourceForge page that you get through a Google search is down, but they do have a GitHub site that still remains and provides a lot of useful information for understanding what should I include, for example, when I'm writing a paper about metabolites. They published a guide to this in 2007 and have done a follow-up study to see how many people in metabolomics are continuing to follow that, that standard for what gets included in the publication. But the group I'm in, the Proteomics Standards Initiative, has also been spending a little bit of time thinking about metabolomics. So the MZ-Tab-M format is designed for reporting quantities from metabolomics experiments. And I really believe that with the support of PSI, certainly, we believe that the Metabolomics Standards Initiative can keep doing the good work that they have been. So certainly, metabolomics has changed a whole lot since I was a graduate student next door to Gary Suziak's lab at Scripps. So there's a whole lot that's been changing over the last 2 decades. And there's, you can only imagine that because mass spectrometry keeps getting better, that this technology is going to have more and more applications across the space of metabolomics. You know, I believe that the transition point in any new technology is getting from that place where just the technologists know how to use it to where the biologists who have biological questions they want to answer are using it. And it's quite plain that metabolomics is well into that space where biologists who simply want to answer their biological questions are able to use these technologies successfully. So targeted experiments are brilliant when you want the highest possible sensitivity and the lowest variance in measurements. And this is happening because I'm going to personify the instrument for a moment. The mass spec's attention is fixed on just the narrow set of metabolites you've told it are part of its target list. If the mass spec is free to fragment just any ion that's showing up, it's going to produce a great diversity of information. But if you tell the instrument through the method that you want it to focus on these and only these metabolites, you're going to get a lot more reliable a quantity out of it. So I think that's really the magic here. Use a targeted experiment when just a handful of the metabolites are what really matter to you. Yes, maybe your laboratory has access just to an LC-MS-MS, but someone near you has GC-MS available, someone near you has a triple quad. There are lots of instruments out there that can support you. There are lots of people out there with special expertise in separation technologies like CE that should also be part of your mix. And you should not say, well, we're a mass spec lab, we don't think about NMR at all. NMR has a lot to show us. So being ecumenical about which technology is going to give you the best answer is going to give you the best biological punch, I think. Naturally, plants are nature's biochemical factories. And the diversity of the products that they can produce is just on an altogether different scale. So I would definitely encourage anybody who's got an interest in metabolites to think about what we can learn, for example, about crops or about the pathogens that afflict crops. And that said, plants in general have all kinds of fascinating biochemistry that we need to know more about. So I would definitely encourage you to think about how the technical skills you picked up, say, in a human metabolomics lab can be applied in another direction as well. All right, and with that, we are through. Thank you.